still on Mendel's work of inheritance, or work in genetics. And the last time we saw what he did, some experiments he carried, and what he actually used, that is using the green pea. Why he used the green pea, we were able to see that. And after that, he came up with the method used by Mendel so as to prove his beliefs. Method proved by Mendel in his experiments. He used two major methods. The first one we said is monohybrid. The second one we said is dihybrid. Then under monohybrid, we realized that he crossed two different plants at a time, which differs in pair of contracting characters, that is tall and short. And when he did the two, when the offspring came out, all of them became tall. When he did the tall, all the offspring were tall. When he did that of short, I think we explained that, and we drew the genetic drafts. So when he did that of short, all of them became short. So that one he called them the first filial generation. And he did not actually stop them. He found out that the planted tree, the one he did, he first of all did artificial crossing, whereby he collected a pollen grain from a tall tree and the stigma from the short one. Did that say something? Remember, he has already carried out an experiment using the very tall one, and also the short one. The offspring all tall, the offspring all short. Now, in order to find out what is happening, bringing the two, so he took the pollen grain, the mature pollen grain, from a tall garden pea, and the mature pollen grain stigma from a short one, and he carried out self-pollination. You know, one of the reasons why he used the garden peas is that they can pollinate themselves. But this one, he decided to do the pollinating using artificial one, like he called it. He pollinated the two together. And once he did that, he has to use a cellophane and cover it. Why? To avoid any birds that might want to come and interfere and bring about normal pollination. So he had to close them using until. So by the time the thing came out, he found out that some of them were tall. Virtually all of them were tall, but not as tall as the tall green pea, and not as short as. So that one, we find out that they were all heterozygotes. So that he called also for the one he did, the first filial generation. Now the second one, that's bring about with what he did in his own, this is called the hybridization. He said he planted tall, the other one he used the seed, the other one he did it by himself, and we have already did some crossing where we used the homozygotes tall plant, green pea, and the homozygotes short. And coming up, we have all heterozygotes in as much as they're all tall. And that one leads to his first, which assignment I gave you, his first theory. Mendel's first theory, after what he did. Using, I repeat myself, I said the first one he called monohybridization, whereby he used the tall tree, he planted those ones, their seed, and the short one, and they all become full tall, full short, homozygotes. And this time around, he carried out using the seed for the two, he still gave it. So now he pollinated the flowers by himself in artificial form. So with that few experiments that he did, that's what led to his first theory of inheritance, Mendel theory of inheritance. And I asked us to go and find out what is it. People mentioned the name, and I need somebody to state it for me. What do you say is the first name? Huh? Uh-uh. No. First law of inheritance. First law of inheritance called what? Law of segregation. What does it state? Law of segregation. Let me see your notes. I asked us to go and read it too. Because you were giving me. I asked us to go and read it. Law of segregation. I asked us to go and read it. Finally, what does law of segregation state? This is the first thing he did. This is parents. Parents gamete. The offspring. So in this case, we find out that, what do we note? That the genotype is what? What's the genotype? All tall trees. And the phenotype? All true shape. Where?
Tree.
Thank you. All tall plants. This one is too. All tall plants. Let me use the word plant. So this is all tall plants as well. But there are heterozygous. So we still find out that Mendel had a crossing of the two from the first filial generation to obtain the second filial generation, which we said he did by himself. Okay, this is the first one. I know you have already done this. Chevy?
Yes,
That is for this first filial, and that result to second filial generation crossing. During word marking, if you're given this is a tick, this is a tick, and that is 2 marks. More especially to have a cross. This is a half tick, this is half tick, this is half tick, this is half tick, that is 2 marks. And this is half tick, half tick, half tick, another 2 marks. Then, if your tick is correct, half tick, half tick, half tick, another 2 marks. That is how many marks then? 8 marks. Then if there's not a question on the number of genotype and the phenotypes, altogether, it might get up to 12. And that might be the homework. Or they ask you from the question, we'll get, at least by next week, we'll be able to get some past questions to treat so that you not take up by surprise. When you are asked, maybe you're getting your answer when nobody gives you the first generation, the first filler generation, and you are given the second filler generation. You have to generate the first filler to confirm what you have in the second filler generation. And the total mark there might just be 20, just from the crossing. Now let's look at from what he did like this. Now looking at this, in terms of genotype, we have 1:2:3. We have 1 tall, which is homozygotes, 1 not too tall, which is heterozygotes, and 1 very short, which is homozygotes, giving us a ratio of 1:2:1. But if it comes to phenotype, phenotype has to do with physical appearance. We'll have 3 tall and 1 short. And when it now comes to the ratio, it is 3:1. Is that clear? So from this thing Mendel did, is what we call monohybridization. So from this work he did, he brought out the first law of inheritance, which he called law of segregation. And what does the first law of inheritance states, which is the law of segregation? Let us write, since we could do further. Is that a past question? So with the above, with this Mendel came up with his first, came up with this, that is these structures you have written. Mendel came up with his first law of inheritance. If you have done this in your notes, and I said with this, Mendel came up with his first law of inheritance, which he called law of segregation. Law of segregation. Law of segregation. This law states that the characteristics of an organism, are controlled by genes. The characteristics of an organism are controlled by genes, which occur in pairs. This law states that the characteristics of organisms are controlled by genes, which occur in pairs. And such pairs of genes, only one can be end. E-A-R-N-E-D. End. You end it. In such pair of genes, only one can be end in a single gametes. Only one can be earned in a single gamete. Full stop. We can also state the law in this way, depending on the one, the English it seems to be. The law can also be stated thus. The law can also be stated thus. THES. Genes are responsible. The law can also be stated thus. Genes are responsible for the development of the individual. The genes are responsible for the development of individuals, and that they are independently transmitted. I repeat myself. I said the law can also be stated thus. Genes are responsible for the development of individuals, and that they are independently transmitted from one generation to another without undergoing any alteration. Without undergoing any alteration. Without undergoing any alteration. First stop. The first one states that characteristics of organisms are controlled by the genes which occur in pairs, and each pair of gene, only one can be earned in a single gamete. And the same can also be stated that the genes are responsible for the development of characters, and that they are independently transmitted from one generation to another without undergoing any alteration. Now, when you look at the first law of this inheritance, we find out that Mendel deduced the law from the idea that the disappearance of the recessive characteristics in F1 generation and its appearance in the F2 generation. Did I say something? No. Did I say something? Now, if we have this, with this first one, you find out that this short tree plant disappeared. In the first one, when he used the pure homozygous tall, homozygous short, after coming to the offspring, there is no homozygous short, although there is no homozygous tall, but all of them are tall. None is as short as. So when it now came, which means the shortness is recessive, you were able to look at terms. Can you remember? Used in, I will set the recessive genes are those genes that does not show in the physical appearance, but they are within or inside the individual organisms, which may appear in the next generation. Did I say something now? So from what he did, he deduced his first law from what he carried out in order to that occurs in monohybridization. The one that disappeared, that did not show at all after the crossing, reappeared in the second filial generation. And what is that reappeared? Homozygous short. So from there, he deduces so that no matter what is being inherited from generation to generation is controlled by the gene and also transmitted from parents to offspring. Is that clear? Yes. So it was during that first method. Remember we said he used two major methods. One is monohybrid, the other is dihybrid. So let's look at the second one, which is dihybridization. Do we need to write this thing? I said no. Mendel deduced the law of the idea that in the first filler generation, you need to write it down. I've explained it, you need to write it down. So I'm able to remember. We should continue. So now I'm talking to human beings. Hybrid inheritance. Dihybrid. Dihybrid, sorry. Dihybrid inheritance. The first one we saw was monohybrid inheritance. Please, let's write that. Mendel also carried out several experiments in which he crossed plants which differed in two pairs of contracting characters. Which differs in two pairs of contracting characteristics. I repeat myself. Mendel also carried out several experiments in which he crossed plants with different, which differed in two pairs of contracting characteristics, such as seed shape. He uses seed shape. For example, round seed, such as seed shape, and then seed color, with seed color, such as seed shape, in bracket round, and wrinkled, such as seed shape, round, and wrinkle, and seed color, yellow and green. Now these are two characteristics, two pairs of characteristics. Remember, he used garden pea because of different things. He observed that some of the seeds are round and wrinkled, some of their flowers are red, some are yellow, and some are what? White. I like to use them. So this time around, when it comes to double pairs of contracting characters being shown in their characteristics, so he decided to use this plant. You use them. Some of these contracting characters have red, they have yellow shape, and some have wrinkled shape. When we say wrinkled, it's wrinkled like this. That is the seed. Some are pure cycle, and then some might be yellow. The same seed or two contracting, some are yellow and some are. In that case, they might say to use red and yellow, and then we'll have wrinkled and green. So you have two pairs of contracting characters, and two of them coming to me. That is the double. Two pairs. This one was a pair of contracting characters. You remember now? And remember the genes, they appear in pairs or chromosomes. So this one was a pair, but these ones are two pairs. That is what we have there. Using the round seed and the wrinkled seed. The seed color yellow and green. So probably it can be represented in this form. So let's go. Contrasting characteristics such as seed shape in bracket, round and wrinkled seeds, and then seed colors, yellow and green seeds. Full stop. He called the whole setup. He called the whole setup. He called the whole setup as thy hybrid inheritance. He called the whole setup as thy hybrid inheritance because pairs of contrasting characters are involved. Because pairs of contrasting characters are involved. That he used two pairs of contrasting characters. So we said dominant allele, dominant allele in terms of this is seed color. We have seed color. Then we have seed shape. So in terms of dominant allele and its expression, we have Y for yellow. For seed shape, we have R for round. Now, recessive. Recessive and expression. Recessive and expression will have Y here for green, and we'll have small r for wrinkle. So this is the same seed where you have this, and this is the same shape where you have these. There are two contracting characters. There are two contracting characters. So let's go. Paragraph. In crossing house seed color. In crossing, house seed color and seed shape are transmitted from generation to generation. In crossing, house seed color and the seed shape are transmitted from generation to generation. So we're trying to cross this. We have crossing pure. What do I mean by the word pure? Crossing a pure stalk of pea plants. Crossing a pure stalk of pea plants bearing RR which is yellow with a pure, I wanted you to tell me before I wrote it, with a pure stalk of pea plant. God bless you, homozygous. Please, when you hear the word pure, because at times in the exam they might not tell you, they might not use the word homozygous. I hope you understand what I'm saying, especially when it is pure, when it is homozygous. Heterozygote, they don't have any other words than heterozygotes. Am I saying something? Although they might say impure, that are not pure. When they say that are not pure, just know they're talking about heterozygotes. But when they say pure stock or pure fruit or pure flower, they are talking about homozygotes. Are you getting what I said? Please. On myself, a rat with a pure breed. A rat with a pure breed. And then you know that the breed has their homozygotes in terms of their color. Please, my writing, this is stock. Stock of pea plant. The pure stock of pea plants bearing wrinkle. Wrinkle. What does this one stand for? Wrinkle. What does it stand for? Wrinkle. Forget about R now. Forget about the R learning. What does it stand for? Wrinkle. God bless you. Shape. We are bringing in two contracting characters. That is shape and color. Did I say something now? Yes. You're doing your eye kuli. You better not do your eye kuli. This is not way to do eye kuli. Because this monohybrid, that hybrid, you can only get the answers when you ask. We'll still look for questions in the past question by God's grace next week. That'll be next week Friday. Because we are using our next week Thursday for exam. Next week Thursday, Friday for the SF exam. Although it might be short. Just a short question. We'll finish, we'll continue. I want us to get this into CES link. Because apart from next week, the upper one, although it will continue until the practical. The practical will be the last day for biology. That day of practical and biology will be the last one. We might not spend up to like 2 hours. Am I saying something now? Have you started your own exam? So you combine the two with the SF next week. Okay, so bearing wrinkle. Please, whatever be it you use in the pure, whether be a pen, you might decide to, because you might not be given the element to repair you deduce your own. They might decide to say height, and you decide to use T, or you might decide to use B. Whatever be it you use for the two parents, if you use capital B, the other one you will use small b. If it is heterozygote and you decide to use capital B and small b, and if the other one is homozygote, you're using small b. You can't use B even if they say white rats and black rats. And you decide because they say white, let me use white, W for white, and black, let me use B for no. They are no longer of the same species. Am I saying something now? So you use the same thing you're using to represent the chromosomes, that is the alleles, on what you use on the two parent organisms. Am I saying something now? Please take note of that. Okay? Wrinkled. Come on. And green color. Apostle. Wine seeds. Result in offsprings. Have you finished writing? I repeat myself. You've already started writing, or you didn't write it. In crossing a pure stalk of pea plants bearing ground. Oh, I didn't finish it though. Bearing round, R, and yellow. Bearing round, LL, comma, yellow, comma, with a pure stock of plant P, bearing wrinkled and green, color, did you get it? I hope perfect. I should repeat myself. Are you with us? Results in offspring that have only round yellow seed. That is seed. Results in offsprings that have only round yellow seeds. Don't have parents, round, yellow, crossing with wrinkle, green, wrinkled green, don't have offspring. So let's assume we have this resulted to all round yellow. So when you cross this, we say crossing a pure stock of pea plants bearing round seed with yellow-green color seed. With a pure stock of pea plants bearing wrinkled with green color seed. It will result in offsprings that have all round yellow seed. That is what we have there. All round yellow seed. Why? This is because, you can write down, I said why, and I'm telling you the reason. This is because the homozygous parents, this is because the homozygous parents produces two kinds of gametes. This is because the homozygous parents produces two kinds of gametes, RY and RY. Two kinds of gametes, RY and RY, and all their various combinations, only give rise to offsprings that are heterozygous. With all their various combinations, only give rise to offsprings that are heterozygotes for both characters. Correct. Now, give rise to heterozygotes that are heterozygotes. That's it. They all give rise to offspring that are heterozygotes. Stop. This only the expression of dominant alleles. This only the expression of the dominant alleles are seen in first filial generation, F1 generation, are seen in F1 generation. First paragraph, each member of the F1 generation undergo meiosis. Each member of F1 generation undergo meiosis to produce 4 kinds of gametes. Each member of the offsprings, or each member of the F1 generation undergo meiosis to produce 4 kinds of gametes, and they are RY capital, RY capital, so the F1 generation, each of them undergo true meiosis and produce 4 kinds of gametes. And they are round, yellow seed, round, green seed, wrinkled, yellow seed, round yellow seed, round green seed, wrinkled yellow seed, and wrinkled green seed. So in the first filial generation, it was all of them appeared to be round, wrinkled, yellow, green. So they now undergo meiosis and produce 4 different types or kinds of gametes. We now have round, yellow, round, green, wrinkled, yellow, and wrinkled, green. Is that clear? Now we're now going to have them cross together, and they cannot do this alone. We'll have to use another method of crossing, which is called Punnett crossing, in order to get the full number of offsprings and their generations. Did I say something? Remember we said a pair of contracting characters. If this what was deduced or what was found after crossing this pair of contracting characters, 2 pairs of contracting characters, that Mendel used in deducing his second law, and what they call law of what? Independence as subject. Law of independence, because they subject the characters and sets it. During this process, it is noticed that the alleles for 2 characters are independently distributed. During this process, it is noticed that the alleles for two characters are independently distributed or assorted. Are independently distributed or assorted. Independently distributed or assorted. Full stop. Therefore, if member of first generation, therefore, if member of F1 generation are crossed among themselves, the F2 generations so produced will show, I repeat myself, therefore if members of F1 generation are crossed among themselves, comma, the F2 generation so produced will show 4 phenotypes and 9 genotypes. Will show 4 phenotypes and 9 genotypes. So let's look at this. This is the parent. So we're trying to say if you bring the round yellow seeds together with this, remember we have this already, with this and wrinkled, this is round yellow, this is wrinkled green, and you bring out the gametes by meiosis. So when they are crossed, that is the fertilization combination, we are going to have all of them. That is what we say 4 genotypes, 4 phenotypes, and 9 genotypes. So this is what we are going to have. If we want to tell ourselves, how did we arrive at this? That is what we're now going to use. Punnett square. This question has come out in WAEC, and you are asked to do it using 2 pairs of contracting characters of a round yellow seed and a wrinkled green seed. How many of us have seen that question? First question? You don't understand it. Now with this, that was how Mendel was able to deduce his second law of inheritance, which is law of independent assortment. They started assorting themselves because some of them, the green, round green color, wrinkled yellow green color, some of them were recessive and they begin to appear. But when you have 2 contracting characters, 2 pairs of contracting character, already we know that the genes, they appear in pairs. When you now have 2 pairs, that's making how many? 4. So each will have 4. How do you go about it? And it also shows most of the time an individual. Because when it comes, apart that he used plants, when it comes from generation, it just recently I got to know that the twin sister is not as fair as she is. And the twin sister might marry somebody of her own color and still have a child of this color. And people will be wondering, how? Because I've seen one. The 2 of them are dark. The whole children are as fair as he is. So they started tracing back from the grandparents, the great-grandparents, bringing the two, and the recessive begin to reappear. And remember that what brought about the first law of Mendel. Did I say something like this? What is no longer crossing, but to bring it in a square form to make it easy. You can use Punnett square where you have a pen as well. Can I clean this?
Yes.
Have you written this?
No.
You know what writer? But you wrote that sentence. That will make it easier for you to understand. Even I cannot clean it.
Yes.
Pleasant to say you did not write it. Okay, somebody said no.
No, you can clean.
Can you remember what we had there? All of them were what? All rounded. Okay? So that is what we're trying to bring out here. So what you have here, you bring it up here. You bring this as well. You bring this as well. So let's go this way to make it easy for us to understand. You know, if we bring it out and begin to cross like this, it might not be easy for us. You understand? So we'll have to use this. So if you bring this, we are going with this one now first. This happens to be the male, this happens to be the female. So if you are bringing this, we'll have this. I'm even supposed to draw it. If we are bringing this, we'll have this. Are you following? Akira, are you following now? Okay? If you are bringing this, we'll have this. If we are bringing this, we'll have this. So we'll continue with this now. If we are bringing this, we'll have this. It's like I'm missing something. This. Okay. round. Okay. So let's number it. I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 offsprings. We have 16 offspring. How many of them do we have as round yellow? Which one is round yellow there? Check now. We have 1, 2, 3, 4, 5, 6, 7, 8. How many there is round yellow? How many is round yellow? How many? 9. Okay. What are the numbers? It's used to determine genotype. Genotype of the F2 generation. Okay, let's see. Round yellow. You said there are many. 9, shall we? You said there are 9. What are the numbers? 1, 2, 3, 4,
9,
4, 9. What of 5? Okay, 5 is there. What of 7? What of 7? And number 7. It should be capital now. Okay, not this one. Which one? Number what? This 7 now. Small rectangle. Alright. Is 7 not there?
15.
Okay. Which other number? 9. Which other number?
10.
Huh?
10.
Are you sure? Are you sure? Are you sure?
Yes, ma. Why not? Like them so that it's easy.
No. How do you collect in items?
Put the small letter R.
We're using this the main. If you're crossing this, oh, start talking about sex determination. This one holds the whole thing. This one is the main determinant. What will happen? And that is why you see them. Or at least you know, for human, SY is for male. And this is for female. Which other number?
10 is there. Ma, 10 is there.
You told me 9. So, how many do we have? 1, 2, 3, 4, 5, 6, 7, 8.
Number 10.
Number 10. Okay. And you were not sure of yourself. I wanted to give you 200 naira. So have round yellow.
9, 13.
Everything 13, okay. And then 13. So have 9 round yellow. Would have shown it. What of green? Round green? Yes? How I got this? Okay. They are all heterozygotes, individual, and they form 4 gametes. So we're now bringing them together to know how many. This one is what? Round yellow. This one is what? Round green. This one is what? Wrinkled yellow. This is what? Wrinkled green. Do you understand it? Now the one at the back is always the recessive part. So we're looking at those ones that are physical. You know what is dominant? You know dominant? Good. That is the outward appearance dominating the one that is like her fair color is dominating the dark color inside of her. Because she inherited it. It's inside. It's recessive. It might show in the second generation of her parents. Did I say something now? In your own, your daddy and mommy is very dark. Your grandparents, are they very dark? They are very dark. Both sides. And they showed on your side. But if you marry somebody like her, you might not have an offspring that will be as dark as you are, as fair as she is. That might manifest following the first law. You were not here. We will talk about the first law of Mendel. Were you here? Not sure. You are attending the other side of lesson, shall we? You don't attend anyone. School on. Your school is having lessons. I mean the afternoon lesson. And you're supposed to be here. That's okay. Combination. Combining the two by meiosis. That is mating now to bring up the offspring. Is that clear? So when you bring this is the matching one. You write this first and write this second. And you have a pure stock. You come to this one. Because you still use this for all of them. Is that clear? You come to this one and this one. It is not pure. But this is round yellow. And this is round green. This is now recessive. This is dominant. Are you getting it? Are you following? And this is round yellow. The first one here is round yellow. When it comes to phenotype, we'll put all of them at the same point because we're talking about physical appearance. Is that clear? But it's not as yellow, green as the first one. This one, green has joined it somehow. So it cannot be pure green, pure yellow. It might be lemon. Are you following? Do you know lemon fruits? One of the citrus fruits. Lemon. You know we have lemon that are pure yellow with a smooth round body. And we'll have lemon that are lemon in color, which means they are not really pure yellow and they are not really green. Still, and the body is smooth. Then we'll have lemon that are pure yellow, but the body is rough. Have you ever seen that? And you have the one that is green, and the body is rough. One of the one inside Baptist Girls College is pure green with serious roughness. that doesn't mean it has spoiled. You know where it is? You don't know. But you know where you have your palm trees. You know where that German mango is. Around that there is lemon orange tree. But you see different types. If you are visiting all these people selling it, I hope you understand what I'm saying. You see round one that looks smooth. You see round one that is pure, yellow. You see round one that is lemon in color, light yellow. You see one that is real leaf green in color, with a rough body. I hope you understand what I'm saying. Yes. So that is what Mendel saw in the garden pea plant. Different varieties of size and color and shape. He decided to use them for his experiment to be able to prove what he's talking about in terms of inheritance or genetics. Shoyeni, I'm asking you. You want to ask a question?
No.
Okay. So this is what we have. This. You tell me how many wrinkles you know, how many wrinkles. As it is from this crossing that Mendel was able to deduce its second law of inheritance. I gave us an assignment, none of you did it. And what does the second law say? Independent assortment. Please, can you write it down?
Yes.
Let's write it down. The second law of inheritance.
Yes.
Second law of inheritance. The law of independent assortment states that during gamete formation, the way one pair of allele during gamete formation, the way one pair of allele states that during gamete formation, the way one pair of allele form a given character distributes itself in the gamete, does not affect the way other allelic pairs distribute themselves. Does not affect. I repeat myself, the law of independent assortment states that during gamete formation, the way one pair of allele for a given character distributes itself in the gametes does not affect the way other allelic pairs distribute themselves. It can equally be stated in this form. It can also be stated in this manner that each character behaves as a separate entity or unit. Each character behaves as a separate unit and is inherited independently of any other character. So we'll diagonalize the Punnett squares when we come tomorrow. So we'll have 9 genotypes, like you said, 4 homozygotes and 5 heterozygotes, and all these lead to the second law of inheritance. Behaves as a separate unit, and it is inherited independently of any other character. 7. Inherited independently of any other character.